Greetings, ladies and gentlemen. In this video, we are going to continue looking at this topic, nutrition in the plants. Okay, so peradventure, they are new followers. LS online tuitions are demarcated into two parts. So the first part covers lesson. So which means 50% of LS online tuitions covers lessons, as we can see. So the WhatsApp number is also included on the screen. So just in case you are interested, you can use the number below. Okay. Then solving past papers also covers 50%. Solving past papers also covers 50%. So far, the feedback from my online learners or online students is awesome, as we can see. The feedback is positive. The feedback from my online students is awesome, as we can see. The feedback is good. Okay, so LS online tuitions are conducted in the WhatsApp group where pre-recorded videos are uploaded at 20 hours. So the duration of each pre-recorded video is 40 minutes. So you are going to see the timetable. You are going to see the timetable. So don't worry about asking questions. Don't worry about asking questions. Learners are allowed to ask questions. I'm always at learners' beck and call. So just in case you have a question, you are free to ask. Okay, you are free to ask. So the price is 150 kwacha. The price is 150 kwacha, and the subject is uh, biology. The subject is biology. So here is the timetable. So each pre-recorded video is uploaded in the group at 20 hours. Okay, at 20 hours. So you can download this video at any time. At any time, you can download it at your uh, time of convenient. Time of convenient. Okay, so we we start. We continue looking at nutrition in plants. So today we are going to look at how we can determine that photosynthesis has taken place, or how we can know that photosynthesis has taken place. So to determine whether photosynthesis has taken place, the leaves of plants are tested for starch. So the presence of starch is an evidence enough that photosynthesis has taken place. So the steps involved in testing a leaf for starch. So step number one. So here is step number one. Boil the leaf in water to kill the protoplasm and make it permeable to iodine solution. So what is protoplasm? So protoplasm is the combination of cytoplasm, nucleus, and other organelles found in the leaf. So the first step, we are going to boil the leaf so that we can kill this protoplasm, so that we can make the leaf permeable to iodine solution. Okay, so this is the first step. So as we can see, the experiment has been set up. We can see the beaker, we can see the boiling water, we can see the green leaf, we can see the stand, and the spirit burner. So leaf is being boiled in water. Step number two, we are going to say boil the leaf in alcohol using water bath. This is to extract the chlorophyll. So after boiling the leaf in water, we now need to boil the leaf in alcohol. Why are we boiling the leaf in alcohol? We are boiling the leaf in alcohol so that we can remove chlorophyll. Why are we removing chlorophyll? So that it does not interfere, it does not disturb color changes. So we are removing chlorophyll so that it does not uh, disturb or interferes with the color changes when adding iodine solution to the leaf. A water bath is used because alcohol is flammable. So what does it mean when we say alcohol is flammable? So alcohol is most likely to cause fire. So any slight provocation can lead to fire. However, the alcohol also makes the leaf brittle. So brittle means hard. So alcohol also makes the leaf hard. So this is step number two, boiling alcohol, boiling the leaf in alcohol in order to remove chlorophyll. So we have seen that chlorophyll may disturb the changing of color or may interfere with the changing of colors. Step number three, we are going to say place the leaf in warm water to soften it. Why are we placing the leaf in warm water? So we are placing the leaf in warm water so that we can soften it. So this is step number three. This is step number three. Okay, step number four. Step number four, we are going to say spread the leaf on a white towel and add iodine, drops of iodine solution. So this is to test for starch. In the iodine, so if the iodine solution stains blue black, starch is present. If it remains yellowish brown, starch is absent. So we have seen two criterias. So if the leaf turns blue black, then we are going to conclude that starch is present. If the leaf remains or if the leaf turns yellow brown, then we are going to conclude that starch is absent. So this is step number this is step number four. Okay, so this is step number four. So we have seen how we can know that or how we can determine that photosynthesis has taken place. So um, during the process of photosynthesis, starch or glucose is produced. So if the test is positive, then we are going to say the process of photosynthesis has taken place. If the test is negative, if the test is showing that starch is not present, then we are going to know that the photosynthesis process has not taken place. Okay, so this is how we can identify whether photosynthesis has taken place or not through testing for starch. So here is a summarized diagram showing all the steps during the test for starch. Okay, so uh, we are going to pick this question from 2017 paper 3. Paper 3 is a practical paper. Paper 3 is a practical paper. So we are going to answer this question from paper 3. So the question is question number 3. The question reads, a grade 12 learner was provided with a leaf with the white and green patches. So this leaf uh, consists of green and white patches. I'm going to show you, which had been exposed to sunlight for 8 hours. The learner tested the leaf for starch using the following steps. So step number one, the leaf was first boiled in water for 2 minutes. So these are steps we were talking about when testing for starch. Step number two, the leaf was then boiled in ethanol. So the ethanol we are talking about here, we are talking about alcohol. So alcohol is an example of ethanol. So the leaf was then boiled in ethanol using water bath. So step number three, the leaf was then submerged in warm water. The leaf was then submerged in warm water. Step number four, the leaf was spread on a white towel. Step number five, the leaf was flooded with iodine solution on the white towel. Okay, so we are told in the question here, that the leaf has white patches and green patches. White patches and green patches, as we can see here. Okay, so we have we have the leaf here showing white patches and green patches. So this is how the leaf was looking like according to the question. Now let us come back to the question. Question 3A, Roman numeral 1. Question 3A, Roman numeral 1, the question reads, state the reason why the leaf was boiled in water. So I gave the reason why the leaf was boiled in water. So the reason why the leaf was boiled in water was to kill protoplasm and make it permeable to iodine solution. Okay, question 3B, question 3A, Roman numeral 2. Question 3A, Roman numeral 2. Why was the leaf boiled in ethanol? So ethanol, we are simply talking about alcohol. So why was the leaf boiled in alcohol? To remove chlorophyll. To remove chlorophyll. Okay, we shift to question 3A, Roman numeral 3. Give a reason why the leaf in ethanol was heated using water bath. So the reason why the leaf was boiled in alcohol or ethanol using water bath, we know that alcohol is flammable. Alcohol is flammable. Okay. Question 3A, Roman numeral 2. State the color and the texture of the leaf after boiling it in ethanol. So the color will change to brown white. So we are just going to say white. Okay. Then texture, we were told that after boiling the leaf in alcohol, the leaf became brittle or hard. So we are going to say the texture is brittle or hard. Okay, the question continues. Question 3A, Roman numeral 5. Give a reason for placing the leaf in a warm water in step 3. Okay, so the reason why we we placed the leaf in warm water in step 3 uh, is to soften it. Okay, remember the leaf is coming from alcohol and the leaf is hard after coming from alcohol, or the leaf has become hard after being boiled in alcohol. So now we need to soften the leaf. Okay, question 3A, Roman numeral 6. Why was the leaf spread on a white towel in step 4? So it was spread on a white towel so that we can add iodine solution. So that we can add iodine solution. Okay. Question 3A, Roman numeral 7. After flooding the leaf with iodine solution, what color changes were observed in the white patches and in the green patches? So we need to understand that white patches have no chlorophyll. Sorry, patches have no starch, so they will show a negative test. Okay, they will show a negative test, or they will show the absence of starch. So therefore, we are going to say yellow or brown. Then green patch, so the green patch will show the positive test, which is blue black. So green patch will show the presence of starch. Will show the presence of starch. So we have the leaf we are talking about here. So we can see that on the white patch there will be no starch. Then green patch will have starch. Okay. The question continues. Question 3B. Question 3B, the question reads, what term is used to describe a leaf with the green and white patches? So the leaf having green and white patches is called variegated leaves or variegated leaf. Okay. Question 3C, name the raw material necessary for photosynthesis to take place. So the raw materials, last time we discussed about the raw materials required for photosynthesis to take place. So the raw materials required for photosynthesis to take place, we are simply talking about water and carbon dioxide. Water, carbon dioxide, sunlight, these are factors needed for photosynthesis to take place. So therefore, we are going to say carbon dioxide and water. Question 3D, the question leads: what is the importance of photosynthesis to human beings? So as we can see, the question is carrying 2 marks, so we need to provide two uh, importance also. So we are going to say provide humans with food and oxygen. So photosynthesis provide human beings with the provide human beings or humans with the food and oxygen. Question 3E, the question leads: name 2 organs in which the plants store the end products of photosynthesis. So the end product of photosynthesis are stored in seeds and roots. Are stored in seeds and roots. So as we can see, we have answered this question, and this question is carrying 16 marks. This question is coming from practical paper, which is paper 3. Okay, we proceed. We proceed. We are also going to do experiments to show that light is necessary for photosynthesis. So we are going to do this experiment to show that um, light is very important during the process of photosynthesis. So to carry out this experiment, the first step that we are going to take, we are going to discharge. So to discharge is to make sure that the plant loses the starch. So we are going to discharge by putting or we are going to discharge a well watered potted plant by placing it in the dark for at least 24 hours. 24 hours, we are talking about a, a day. Okay. During this time, all the starch present in the potted plant is used up. So the plant will lose the starch. Then number 2, set up the experiment as shown in the diagram. Step number 3, place the potted plant in sunlight for 46 hours. While the plant is in sunlight, draw the selected leaf showing the exposed parts and the covered parts. So we are from taking this plant from the dark room, and this plant has lost all its starch. Okay, now we are placing this plant under the sun. We are placing this plant under the sun. Okay, so we consider this leaf, we cover part of the leaf, and we allow part of this leaf to be exposed. Then in the last step, we are going to name this exposed part and the covered part. So we are going to say exposed part, we are going to name exposed part A and covered part B. Then after 46 hours, we are going to test for start. We are going to test part A and part B. We are going to test for part we are going to test part A and part B. Okay, so we have the diagram here. As we can see, we are going to consider this leaf. We can see that part of it has been covered, and the part of it has been exposed. As we can see, the T-shape has been exposed. The T-shape has been exposed. Okay, so what will happen to the exposed part is that when we test for starch on the exposed part, we are going to have a positive test, or the exposed part will show uh, the presence of starch. Okay, then the covered part will show the absence of starch. So we can now say that light is very important in the process of photosynthesis. Let us also answer this question that is coming from 2019 paper 1, which is question 8. Question 8, the question leads: the diagram shows a variegated leaf. Remember, a variegated leaf, we are talking about the leaf having two colors, green and white, or having two parts, two patches, green patch and white patch, like we saw in the previous diagram. Okay, so the leaf was exposed to light for 4 hours and then tested for starch. With legion will turn blue black. So we have seen that here we have the white part or white portion. So this white portion will show the absence of starch, and the covered part will also show the absence of starch. The green part will show the presence of starch. The exposed part will also show the presence of uh, starch. Okay, so therefore our correct answer here, our correct answer is C. So 2 and 4 will show the presence of uh, starch. Okay, will show the presence of starch. So in this video we are going to end here. We are going to end here.